posted on later. All right. So, hi everyone, I'm Russell. I already kind of introduced myself yesterday. This is day 2 of the Ecology and R web conference series. Super thankful that everybody is giving us their time to talk about their research and their R skills and everything. Sorry about the technical difficulties. We had a little bit yesterday with people spamming the link on other people's page, which actually turned out to be like a virus or something. So I did not see that coming. Today, I guess we're not going to have that problem because it won't let us post it at all. So the speakers are going to talk, and then I'm going to have the Q&A dialog box open. And whenever they finish their talk, you guys can ask some questions. And if they can see it, then they'll answer as they go on. Or if they can't, then I'll read it out. All right, so I will let Nicole take over from here.
Okay, so I will go ahead and try to share then, I guess. This is my first virtual conference, so I'm kind of excited. Let's see how smoothly this goes. I do teach virtually, so it's kind of the same process. It's very clunky, you know, it's like, let's step by step, share, start the slideshow. Okay, are we good? Russell, maybe you can give me a thumbs up and let me know that I'm sharing correctly. It looks good.
Yep, everything looks good.
Okay. All right, well, I'm excited to talk to you guys today about applying dynamic time warp clustering to dive time series, and I'm applying this to a highly migratory species, one of our characteristic megafauna, leatherback turtles. And I am Nicole. I do go by Nikki, and I'm a PhD candidate at University of Maryland Center for Environmental Science, as well as College Park. So first, I just want to introduce what dynamic time warp clustering is. And I think that this figure kind of does a nice job of summarizing this. I definitely reference people if you're interested in this kind of technique to check out this paper I cite in the bottom, Lee et al. 2020, because they have some nice detailed descriptions of this. So, you know, just thinking first about clustering techniques. Generally, clustering techniques have kind of relied on using Euclidean distance. It's basically a type of multivariate data analysis method where you're going to try to group objects into k number of groups if you're using k clustering. And you're dividing objects into clusters based on their similarity. So you're trying to maximize that similarity by using some kind of distance measure between your points. So classically, people have used Euclidean distance, and this is where you're going to sum the linear distance at some point in time. Now, the problem with Euclidean distance is that you can sometimes lose information, and you might not be able to actually capture the shape of a time series if they're offset in time or length. So dynamic time warp clustering is a type of K clustering method where, again, you are sorting your clusters into K number of clusters based on their similarity, but you're comparing two time series by changing the comparison time point in time. So it's flexible. And it actually allows for time series that have time lags and for time series of different lengths, depending on what type of dynamic time warp clustering method that you're using. And so essentially you're matching your time series in a direction that is going to be minimizing the distance between them. And so this next figure kind of just walks you through visually if anyone is interested in going back through this on the recording, or if you want to check out the paper and read through this in detail about what dynamic time warp clustering is actually doing, how it's actually comparing and minimizing the distance between time series. This is what it does. Essentially, you're creating a local cost distance matrix, and then you're going to create a global cost matrix, and then find the optimum time warping path between those time series, which essentially is the sum of the distance between the two time series. That is the minimal distance. So now I'm going to introduce you guys to my study system, leatherback sea turtles. These guys are really, really interesting. Here's a picture of one, an adult, and it's feeding on some gelatinous zooplankton. This is what they eat. Leatherback turtles are kind of unique from other sea turtles in the fact that they don't have that hard shell. They are very long distance migrators. They conduct the longest migration of any reptile. they actually have the ability to thermoregulate. They're very deep divers. So even though they're reptiles, they're able to moderate their body temperature. And they're very deep divers and associate with different parts of the water column because they're feeding on this gelatinous zooplankton, which is kind of out in the middle of the ocean, and also in areas where waterfronts come together. They are also endangered. Many populations are endangered, and the Eastern Pacific population in particular is critically endangered. And this Eastern Pacific population is kind of unique. The Eastern Pacific population conducts this migration down to the southeastern Pacific Gyre, and it kind of goes through a migration corridor as it does so. So they will, nest for a period of time on the coast of Costa Rica, and then they will leave and conduct their migration to pelagic foraging grounds for a time, and then return to nest after a while. So this plot on the top right is actually from my collaborator on this project, Dr. George Schindlinger. This is a paper that he did, and it's just showing you tracks for Eastern Pacific leatherback turtles, a bunch of females, a large data set that they had, showing essentially the latitude division between their migratory and their foraging grounds. So they conduct this migratory phase, which is kind of a corridor. And then after that time period, after they pass the equator and kind of head down to the South Pacific Gyre, that's when they kind of disperse into their foraging phase. And during that time, this graph is also showing their dive depth. They're actually associating with different depths. And there's a couple trends that we've already been able to pick out for their diving. When they're foraging, generally their dives when they're foraging are deeper and longer on average than their migratory dives. And they also have a diurnal pattern to their diving. So during the daytime, their dives tend to be deeper and shorter than their nighttime dives. However, they also conduct some really, really deep dives. So, they do have dives for foraging that are deeper, but the really deep dives, the ones that are greater than 800 meters, are mostly during the migration and during the daytime. And these really deep dives are probably for heat shedding, as well as movement efficiency. So now introducing you guys to dive profiles. Dive profiles is a way of illustrating how animals are using the water column over time with their diving. And the shape of a dive profile can actually tell you a lot about their activity and what they're doing during that dive. So, sometimes, like I said, they're potentially using dives for shedding heat. They could be using it for searching for food, for resting, or actually actively feeding. And so for leatherback turtles, dive profiles have been made. Sometimes it's kind of a hand sketch kind of method where you go through each individual time series and you essentially match the points. These points come from time depth recorders. Often for leatherback turtles and other long distance migrators, we're putting satellite tags on these individuals, and you're getting a few positions a day. And then you will also, if you attach a time depth recorder, which is essentially a pressure sensor, that pressure sensor is going to record depth whenever they dive, and it will usually take a handful of points during that dive so that you essentially can sketch a profile of the dive over time. So, people have done this for leatherback turtles, but again, it's been kind of clunky. A lot of times people have used descriptional data of dive parameters, so like summarizing, maximum depth and dive duration for dives, or they've kind of gone through Euclidean clustering methods to try to classify dive profiles in time. They're also generally biased to Atlantic populations. No one's really done this for this particular population, the Eastern Pacific population that I'm looking at. And they're also tend to be biased to internesting individuals. So, and what that means is that leatherback turtles, they spend a period of time nesting, and during that time, they're actually onshore nesting, but then they also will go to the nearby waters and associate with some shallow water during that nesting period for a few months. So that's what we call the internesting period. And it's, a shallower area because they're associating pretty closely with the coastline and returning back to nest regularly during that time period. So it's not really capturing that information, that foraging phase and their migration phase where they're in deeper pelagic waters. So the goal And I did see that you guys encourage memes, so I have put a handful of memes in here for you guys. Hopefully it doesn't distract too much from my points. But the goal of this particular analysis was to use dynamic time warping to classify different dive profiles for diving time series for the Eastern Pacific population of leatherbacks. And particularly, these are all female post-nesting leatherbacks. So they have gone through their nesting phase, their inter-nesting phase, and they're only going into migration and foraging at this point. And so this is the data set. This is actually the data set that was in that Schillinger paper. So for the dive information itself, I had 33 individuals, and this amounted up to over 27,000 dives. So again, you know, it would be almost impossible to go through all these dives and score them by hand about what type of profile they are and try to match the points. So that was a big driver for me to try to apply this method with the idea that it might be fast and might allow me to categorize dives of different shapes and patterns very quickly. You know, generally these dives, each individual has, you know, anywhere from 100 plus to over 3,000 dives per individual. These are pretty long tracks. Again, all these are post-nesting. The dive time series are offset in time and length, which is another reason why I want to apply the dynamic time warp clustering, because Euclidean distance just wouldn't work. These dive time series are not matched in time or space. And these are the type of tags that we use. These are satellite tags. These tags record position, you know, latitude and longitude, as well as total intermediate dive duration. So again, with that pressure sensor on the tags, it's recording essentially 4 intermediate points whenever they dive, and those intermediate points have a timestamp and as well as a depth stamp. So you can get a maximum dive depth and you can get intermediate dive depths. And the intermediate dive depths do actually include the maximum dive depth. So 4 depths total, and then there's a surface time and an end time. And I also had multiple groups here, you know, some different categorical variables that we could think about. I've got 3 to 4 different years. So it's the time span is going from 2004 to 2007. I have night and day categories, as well as migratory and foraging phase categories. The map here on the right is just a quick plug for using this fun R package, Map View. If no one has actually tried this package and you have spatial data, it's really nice. This, of course, is a static screen grab that I took, but Map View in R allows you to plot your data onto a base map. So this is actually like the Esri ocean base map here. So it has bathymetry and, you know, interesting features on there. But it also allows you to zoom. So, and you can categorize your data by color and size and shape and all sorts of things. So here, this is showing you foraging versus migratory phase. So again, pretty similar to that Schillinger paper. We're just showing you where they're starting into that foraging phase. So this is just a quick summary. I actually have a visualization of this next, but if you want the text version of what I kind of did to prep the data, I would say probably one of the biggest challenges I ran into with applying this method in the particular R package that I used for the dynamic time warp clustering was that it took a decent amount of data prep, both beforehand and then after, to try to get the data back binded to my data frames in a way that I could start plotting it. So I'll talk a little bit about that. But essentially, to prep the data, you know, I got the data, my raw data time series. I had, you know, dives for each individual, but the dives didn't actually have the beginning and end dive depths recorded. So I had to add those. You know, they're at 0 depth when they come to this, when they're starting the dive and when they're ending the dive. So I had to add that so that there is those 0 points in the ends. I then split the data set up by each individual dive and then had to organize them by time. And then I create a sequence of timestamps from 0 to the maximum timestamp for each dive and put that in data.table format. I labeled each timestamp with a dive ID, and then I then merged that back with the original timestamps. And then I used the impute TS R package and their NA interpolation function to essentially fit a straight line between points. Because even though these dive time series are allowed to be offset in time and length, they do need regular points. So essentially I needed points in a regular format for each dive time series. So I had to pick a interpolation or a time interval that made sense for my data, which I decided on for 20 seconds, looking at the minimum dive duration for all of my time series. So the minimum dive that I had was 80 seconds. I chose a 20-second intervals that I would have, enough points essentially to create that dive profile. I'm not really interested in the fine scale points. I don't need points every second. I don't need points every 10 seconds, because with the dive profiles, all we care about is the overall shape. All we care about is the maximum depth they're hitting, the dive duration, and the general shape of the dive, which you can understand by just essentially fitting a straight line between points. So hopefully that makes a little bit more sense as I go through the rest of my slides here. But this is kind of what my data looked like, to give you a little visualization, which, this might be helpful for people if they're going back through this talk and taking a look at it. But yeah, I essentially had to add in zeros into my data. And what I end up with here, the bottom black box, is showing you just the first element of a list. I end up with a list of all of my dives, and each element of that list is a separate dive time series, and they can be different lengths. They don't have to be matched in time, but they do have to be equal interval. So each one of those points are 20 seconds apart. And so this is kind of what the data looks like. This little plot on the side shows you, for like one example dive, the intermediate dive points, the raw data points, which would have come from just the data itself, including the surface and end times here. There are 4 intermediate points. One of them is the maximum point. And then I took, 20-second interval points along that dive time series using the interpolation, the NA interpolation function. So basically just fitting a straight line between the points so that I can compare them. And so introducing you guys to the R package that I used. The R package is called DTW-clust. And there's a bunch of different options for this. So, definitely check out, if you guys are interested in this package, check out the associated documentation for it. And there's a lot of background reading on all the different options that you can choose. But essentially, you're going to fit this function called TS-clust, and you're fitting it to your data, which, here again, that data is a list of all of my dive time series that have now been regulated in time so that there are points every 20 seconds for each one of those series. So the first thing I'll tell you guys that I picked was the distance measure. I picked DTW basic. I'm not sure that I completely understand the differences between DTW basic and all the other different distance measures, but the DTW basic, from what I understand, it uses the DTW distance measure itself and is faster. It's performing these distance measures in C++, and from what I was able to read, there's no reason for me not to pick this particular distance measure. The next thing you can pick is your clustering method, which you don't see that an option. I just took the default here, but there are multiple options for clustering. You can choose a hierarchical clustering method. If you choose a hierarchical clustering method, you're going to cluster your data using a hierarchy. So it's going to go through your data and kind of make different hierarchies of clusters. And you're going to end up with, essentially having a very time and memory complex fitting because it is hierarchical. You can also have a fuzzy clustering method. Fuzzy clustering methods is actually creating this kind of soft partitioning of the data into clusters. And so each data is going to belong to, each time series is going to be put into a cluster, a particular cluster, to a degree. So it's not going to be, it's like, for example, if you had a time series of 4, you had 4 time series and you're trying to fit it to 2 different clusters, time series 1 could belong, like 80% of it belongs to cluster 1 and 20% to cluster 2. Whereas the partitional method, which is the one that I use, the default, partitional is assigning the data to one cluster from k total number of clusters. So each time series can only belong to one cluster. And that brings me to looking at the k, or number of clusters. You also have to tell it how many clusters that you want to fit your data into. So how many groupings that you expect your data to be in. This can be kind of tricky. So I'll tell you guys a little bit about how you might choose a particular K. Another method you can pick is the centroid. I chose the partitioning around medoids, PAM. This basically, it's clustering my centroids, and I'm not sure how to explain this one too much. You're forming a cluster using a medoid, and the medoid essentially is a representative object at the center of one of your clusters. So as you go through your time series, the way that this is to kind of generalize how it's fitting the PAM method, it's going through your time series and it's going to pick a time series that is best representative of that cluster. And then it's going to update it iteratively. So it's going to go through and it's comparing each one of those time series to each other, and it's going to keep updating the centroid until the optimal centroid has been decided upon, or representative object. And for PAM, your final centroid is actually going to be one of the time series from your data. So it's not an average. It's not like the average time series that best fits that cluster. It's actually going to be a time series from your data, which can be kind of helpful. I thought it was really helpful for mine because it actually pulled out, again, time series from my data. You can also, there are a couple other options you can choose. So for example, window size. Window size would allow you to limit the distance that points could be matched upon, which I didn't want because I wanted all of my dive observations to be included. So going into choosing the optimal number of clusters, this is kind of tricky. We say optimal because it's a little bit subjective how many clusters. Really, this is a point where you really want to come into this type of analysis with information about what you expect to see based upon the literature for your species, your study system. And, I chose essentially, I expected there to be any number of 2 to 6 different clusters to be identified, which for my particular research question here, those clusters are representing different dive profiles. And based on the published literature for this species, there are anywhere from 2 to 6 expected different dive profiles that I expect to see for these leatherbacks. And, those dive profiles, of course, are profiles of different shapes. And I'll show you guys what those look like with my results. But you can use these clustering validation indices to try to get a better idea of what cluster would best fit your data. Again, it's a little bit subjective. You do have the option of using an internal or external. The internal is where you're going to be just considering your data itself to try to define a measure of cluster purity. Whereas the external, you're going to be comparing your partition data to a correct one. So essentially, to do the external one, you need some sort of ground truth data. So for example, if you already had data that was clustered based on, you had gone through and observed, like for me, it would be, all of my dive profiles. If I had gone through, a certain number of them and scored them by hand about what type of dive profile, what shape or what cluster they belong to, I could then use the external one to compare and see if, that K is actually correct, if there actually were 3 different dive profiles. There are multiple different indices that are a part of this, and the documentation for this particular package recommended that you could use many of these indices and then compare them with a majority vote based on what they're saying. So kind of similar, if you guys are familiar with AIC and things like that, this is kind of a similar concept to that. So for these different indices, the ones that you would spit out with the CVI function, you want smaller values for the S, CH, and D indices, and you want larger values for the DB, MDB, and COP indices. So I went ahead and went with the majority vote, and as well as let myself be informed by the biology and what I already expected from this based on previous literature. So this is the function that you'd use. You specified 2 to 6 here. So I'm telling it that I'm looking for, I want to, you know, apply these indices to 2 to 6 different clusters. And then this is the code to kind of create a data table that allows you to compare those indice values. So I'm going to go ahead and get into my results here, and I'm sure people will have questions towards the end about methods and things like that and how to apply this to your own data. So let me know. Hopefully I'm doing an okay job explaining things. I kind of just recently applied all of this. So this is sort of still recent. These results are pretty recent. So for my results, I got actually 2 different optimal number of clusters. So actually the DB, the MDB, and the COP indices told me that K equals 6 seemed to be optimal, whereas the C or the CH, the D, and the S indices told me that K equals 4 was the optimal. So I then had to again apply you know, knowledge that I had in my study system to try to decide what K was the best. So I went through and I you know, plotted the different centroids or dive profiles that each cluster had, and looked at them and, you know, thought about how it related to my particular study system, whether it made sense or not. And it basically came up with K equals 4 is the most biologically relevant for this particular system. K equals 6, the centroids were actually didn't look to be that different from each other, and I don't think we're going to be as informative for the ecology. Runtime, I originally tried to fit all of this with 1 second intervals between my points. So I mentioned that I had to regularize my dive time series before putting them into this. I regularized them into 20-second intervals. I originally tried to fit them on like a 1 second interval, which was dumb. And it was, of course, you know, completely bogged down. It was taking like a week to run. And I was like, okay, now I'm going to split my data into multiple groups and try to run it on groups. And, you know, finally, you know, I talked to someone about it and they're like, just break it into larger intervals. And, you know, that essentially brought the runtime down to 2 hours. And I was able to run all of these dive time series at once. I think it fit on about an hour something. So pretty fast to fit over 28,000 time series, and comparing each one of those time series to each other, and then sorting those time series into different clusters. This plot here is showing you guys, if you were to just do a raw plot of the output of the TS-clus function, this is what it would look like. Again, like I kind of mentioned at the beginning, I think one of the disadvantages of this package, the way it is right now, is that it kind of takes a lot of post-processing and pre-processing of your data to get it into a format that's really nice for plotting and for binding it back to your original data, and, you know, et cetera, et cetera. So if you were to just plot the raw object, the object that comes out of that TS-clus function, this is what it gives you, which is kind of, it's really nice if you just want to get a general idea to see if the function worked and see what the output kind of generally looks like, and make sure it makes sense. So each one of these boxes is representing the different clusters. I had 4, and there is depth on the y-axis and time on the x-axis. And each one of those lines that are different colors is an individual time series from my data. And you can tell that you can't even see the centroid here in each one of these groupings, but it's kind of covered. I have a lot of data, so it's really hard to see. So, you know, definitely you want to spend time afterwards sorting and processing your data to make it look nice in a graph or a plot. So for the K equals 4 centroids, these are my final centroids that it picked out for each one of those clusters. And this is actually really cool because each one of these centroids actually matches a published profile from the literature. And so this plot here on the top right is from this Hochschild 2014 paper where they kind of did a review on all of the different dive profiles that have been published for different sea turtle species. And these were the 8 different profiles that have been published for all sea turtle species. And of these, there are about 6. There are 6 that are known that have been published for leatherback specifically. And so these black and orange ones actually kind of match this type E. It's a little hard to see with the time plot here with them all overlaid on top of each other. But, you know, essentially that E profile is going down deep, and then it kind of has like a plateau before coming back up to the surface. Whereas the blue and the green ones kind of match this type C profile, where it's going down to depth in like a V shape and coming back up again. And this is really interesting because these different centroids also are representative of pelagic foraging and feeding. And of course, thinking about leatherback turtles, unlike some of the other sea turtle species like green sea turtles and other ones that spend time going down to the bottom and doing kind of benthic foraging, leatherbacks are hunting throughout the water column. So they're probably going to have less of those profiles where they're spending a long time on the bottom, such as the A, B, and D profiles here. So this is kind of interesting. This kind of matches what I would expect to see, especially for this particular population that unlike some of the other Atlantic populations that associate with foraging areas that have higher densities of prey, the Southeastern Pacific is kind of a nutrient deplete ocean gyre, and aggregations of prey tend to be more spread out. So leatherback turtles in this region have to spend more time searching. So that's kind of characteristic of these different dive profiles. So these next plots are just going to show you it's just kind of repeating that first plot that I showed you, that raw output plot, but a little bit nicer. If I was to plot each centroid over all of the dive profiles that belong to each of that centroid, this is what it would look like. The red is the actual centroid identified for that cluster. So this is cluster 1 and 2 here. And the black there is each one of those time series. Now I had a lot of dives, so that's why it's really hard to see kind of the overall shape of the dives, unfortunately, because there's just so many. And I did plot these in ggplot and use an alpha of 0.1. But even with that, it's still hard to see. Also, it's something to note here is that I plotted these on different scales. They're plotted on different depth and time scales. So you can't necessarily compare just visually for this particular plot, the depth and time for the centroids, because they are on different scales here. I did that so that you could visualize the shape of the centroid over the dive profiles itself. So centroid 1 here, you can see that the bottom of that centroid is at around 40 meters, and centroid 2 is shallower, it's at 20. And this is centroid 3. Really interesting because centroid 3 essentially seems to have captured all of the really deep dives. It's got, you can see that V shape to it. And so all of the really deep dives, anything greater than 250 meters was categorized into centroid 3, cluster 3. And centroid 4 was also kind of a deeper dive. It's got, again, that V-shaped profile, but these are dives that seem to be less than 150 meters. And you can extract, so if you were to go through and take that raw output as well from that TS cluster function, you can go through and extract the clusters. But again, it does take a little bit of post-processing to get it into a usable format for plotting. So this is just some summary statistics of the different clusters and their average dive duration and depth, maximum dive depth. And this is generally what I'm interested in, essentially 3 different things. I'm interested in the average max dive depth, the average dive duration, and the general shape for each one of these clusters. So again, just interested in the overall shape. And so for cluster 1 was the second shallowest. Cluster 2 is actually the most shallow. And you can see that it was around 18.3 meters on average. Cluster 3 was the deepest, as we saw in that plot. And it also had a really large standard deviation because of it capturing those really, really deep dives. And the dive duration, the differences between the clusters was actually a little bit harder to distinguish. It looks like cluster 2 did on average seem to be more shallow than cluster 1. But I have, some violin plots here to kind of visualize that. So for the maximum dive depth, there's definitely differences between the different clusters. There's more minimal differences in the dive duration between these different dive categories. This next thing is just talking to you guys about looking at how accurate the centroids were for categorizing the main dive profile. So again, with this partitional, this PAM method, it's going through all of my dive time series. It's sorting it into K number of groups or clusters. So I chose 4. And then within each of those clusters, it's iteratively updating the centroid based on comparing it to all of the different dive time series in that cluster and trying to minimize the distance. So hopefully what I should end up with is that ending centroid, the general shape of that centroid should be the majority, should be the dive profile that's most prominent for that particular cluster. So I just went through, and there might even be there's probably a better way to do this, but I did just, kind of come up with these results this week. But I went through a random sample of 100 dive profiles from cluster 1, which had 52,000 dives. And I just went through and scored them by their different types. So type E was about 33%, and that was the overall centroid, followed by type F, type C, type D, and type A. And you'll recognize these profiles from that Hochschild review paper that I showed. These are just general profiles that have been described for leatherbacks and other sea turtle species. And I thought this was really interesting because it does seem that the type E centroid was the dominant centroid, which is good. But it was pretty closely followed by type F, which actually makes sense because type F is actually, even if you're just visually comparing them, is actually pretty similar to type E and represents similar behaviors. So again, that pelagic foraging and feeding. And then followed by type C, which also again is pelagic foraging and feeding. And the other types identified were type D and A, which have more of a plateau on the bottom and are more for benthic searching and feeding. So it's kind of interesting. Does kind of follow what I expected to see. So the next few slides are just going through and thinking about the ecological relevance of these different profiles and whether it matches what's been published in the literature so far for this particular data set. And so this is just a bar plot showing you the proportion of each dive profile based on groupings. So again, I had a couple different groupings that I could break this data into. I could break it into foraging versus migratory phase. I could break it into day versus night. And I could also break it up by year, which I didn't do, but it might be something I will do in the future. And so here we can see that the foraging generally had more deep dives. The deep dives were these green and blue profiles. So there was a higher proportion of those green and blue profile dives in the foraging phase, which was to be expected. The migratory phase had more of the orange and black, the more shallow dives. And this matches, again, the literature that I introduced in the beginning, that there are more deep dives during the foraging phase. However, there are the very, very deep dives greater than 800 meters do seem to occur mostly in the migratory phase. Also looking at day versus night, daytime, there wasn't a whole lot of breakups between the two for migratory. There is, that diurnal pattern does differ between the migratory and the foraging phase. So just looking at the migratory phase and the proportions of each type of dive type, they look pretty similar. There does seem to be a few more of those V-shaped profile dives at night during the migratory phase. During the foraging phase, those differences are a little bit more obvious. There were more V-shaped dives during the nighttime phase than the daytime. Now, this particular categorization, of course, is it's just binning them based off of nighttime versus daytime, which was, from a particular time grouping that I chose for what's day versus night, a categorical grouping. So you can also group these on a continuous way. This is actually using like a histogram plotted on top of like a clock face, essentially, to get an idea of what the count of each dive profile was for the hours. And so it's kind of interesting because you can see that the C1, C2, and C4 And C1 was black here, C2 was orange, and the C4 was green. You can see all of those kind of occurred throughout the day, but there was differences in the count at different times of day. All of them seem to have a peak at night, which is interesting. The C2 ones, the orange dives, they had the highest count around midnight as well, and also early AM time periods. So you can see from about 1 to 9 in the morning. And that's kind of interesting. These, of course, were shallow dives, and they also tend to be a little bit longer. And this is probably matching actually what we'd expect to see for their prey. Their prey does have a diurnal migration. If you're familiar at all with that concept in the ocean, essentially gelatinous zooplankton and fish and things like that have a diurnal pattern of migration where they'll travel from areas deeper in the water column to areas that are shallower in the water column at night to feed closer to the surface at night. And so then all the predator species will also follow those prey species to the surface. So I expected to see a high number of these shallow dives at night. Actually, the blue dives are these V-shaped dives, these which tend to be deeper. Some of the really deep dives, they did occur also at night, but they occurred as well throughout the day. And this is also kind of what I expect to see, like these dives in the afternoon. During the afternoon, these turtles are probably doing a lot of foraging and exploration, just exploration, where they're kind of just like scouting out. We think they're probably scouting out where their prey are. They're also doing shallow dives though throughout the day. These shallow dives are them kind of resting. So it's kind of interesting. Look at these. I see someone has raised their hand. Let me see if I can take a look at the chat.
I think they just might be having connection issues and they were having trouble hearing you, but I think it's fine now.
Oh, okay. Sorry. Zoom is so fun. Also, I feel like I'm talking a lot. The virtual format is really interesting. So if anyone has questions, do feel free to ask me as I'm going through, and hopefully I can catch it. So as well as the temporal distribution and seeing if it matches what we expected to see ecologically for this species, also want to think about what the spatial distribution of these dives were. And it is interesting because there does seem to be a difference in the spatial distribution of these dives, and it does seem to kind of match what I expected to see. I'm not sure this graph is the best way to represent that. Again, I kind of just like threw these results together yesterday. Some of these end results, some of these end plots. So there's probably a better way to represent the latitude here and change up the x-axis. But here the x-axis is showing you latitude going from -40 to, you know, positive values. The equator's at 0, of course. And so again, just to remember, they're doing that migratory phase, that like migration corridor in higher latitudes. They also, however, do have foraging areas. They do spend some time foraging up in the higher latitudes as well. There are some upwelling regions. Upwelling in the ocean is essentially bringing nutrient-rich cold waters from depth to the surface, and you know, it essentially creates this this food boom, this production boom that starts with plankton and then brings all the fish and the gelatinous plankton and things like that. So you get a production peak in areas of upwelling. So there are upwelling areas actually in some of those higher latitude regions, which kind of matches the black and orange dive spikes here that we see. And that also makes sense because upwelling, you know, these upwelling areas are bringing prey closer to the surface. So you'd expect to see more of these shallow dives in that area. As well as, you know, we also expect to see some of these shallow dives occurring with the migration phase. So of course, as they're migrating, they're generally having more traveling dives, and they have a few of those really, really deep dives, but mostly the deeper dives are occurring in the foraging phase. And then we see kind of, there's a little bit of a spike of the green, the V-shaped dives, and the blue-shaped dives around the mid-latitudes, as well as towards kind of the lower latitudes. And that also kind of matches with our expectations. This is matching some of the other foraging areas that we think that they're using in these areas. In the South Pacific Gyre, in the middle of the gyre, in the mid-latitude regions, prey tends to be deeper. The nutricline and the thermocline, which are essentially kind of how you think about where the productivity is, are deeper in the middle of the gyre than they are towards the edges of the gyre and at these higher latitudes. And so the prey itself is actually more at depth, we think, and also more scattered. So we think that the turtles are spending more time foraging at depth and more time searching for their prey at depth, which matches these kind of V-shaped profiles that we see in those latitudes. So this is another map. Again, I'm using GGMap. I guess the only problem with this GGMap thing is that it doesn't have my latitude and longitude points on here. But at least it gives you an idea of where you are with respect to the coastline. So these are all my dive points, and they are colored by the different dive profiles. I realize that the blue and green is kind of hard to distinguish here. So I did go through and plot them separately so I can tell you guys what the different patterns are. But you can already see just looking at this that there does seem to be a pattern visually and spatially with these different dive profile types. So again, up at those really high latitudes, you're seeing more of those shallow dives. Then you see some of those V-shaped dives. And around the equator, you're seeing more of the shallow dives. The equator also tends to have some productivity closer to the surface, some prey close to the surface. So they again might be doing more shallow dives in that area. And then once they hit below the equator, they encounter around -8 degrees. They encounter their foraging phase areas, and they're just starting to disperse out into the gyre, into that open ocean area. And they have a lot of those V-shaped profiles because they're spending a lot of time searching and feeding. Then as you get further down into lower latitudes, there's something called the subtropical convergence zone, which is essentially an area where ocean fronts are coming together, water masses are coming together, and that actually tends to congregate these gelatinous plankton together and creates higher densities of them. So that's where you again see some more of those shallow dives. So let's see if I can click through here. The next couple slides, I just wanted to compare the distribution of these dives with some published data from Dr. George Schillinger that's showing some of the environmental covariates that we see in this area. So chlorophyll A in the ocean is our measure of primary productivity. Of course, wherever you see higher primary productivity, which in this case on this map on the left is represented by dark green colors, wherever you see higher primary productivity is where you also expect to see, more of these sea turtles and more of, fish and other predators and things congregating to that area. So as you can see, the middle part of that gyre, once you're around like from 10 to 20 something south latitude, it's kind of dead out there. There's not a lot of productivity. We do think that there's still prey out there. So they are still feeding, but the prey is not as dense. So it's kind of spread out. So again, they're doing more of that searching and exploration, probably a lot more of those V-shaped profiles. And then you can see there's some upwelling areas close to the coastline. That's where that dark green is. And that kind of matches where we see the orange dives here. Upwelling again is where you're congregating prey close to the surface. And you can also look, this is a map also of their travel rate. So showing where they were going slower and where they're moving faster along these areas. So, they're spending time in the gyre searching, which is why their speeds are a little bit slower in these areas. The speeds are represented in blue and green here for slower speeds. So they're moving slower in the gyre because they're spending a lot of time searching. That migration corridor is mostly in red. They are doing a little bit of foraging during that migratory phase, but they're mostly just, moving through those higher latitudes. And once they get down to the really low latitude, so below 30 south, that's where that subtropical convergence zone is. And, even though this area doesn't have as high of chlorophyll, which, if we go back a slide, you can see the chlorophyll is actually kind of low there. The surface productivity is not high, but the ocean fronts are coming together and likely congregating gelatinous zooplankton. And so that's why you see, still quite a bit of foraging at those particular areas. And they're also spending time in those areas. They're moving slower. So hopefully that wasn't too much to throw at you guys. Again, it's still a little bit rough. I need to refine some things on this, but I'm really excited about the results so far. Obviously this method did work successfully to identify dive profiles of different shape and depth for this species and this particular population. Not only that, but it was really fast and it was efficient. I didn't have to go through and hand score any of them. The centroid for each cluster was the dominant dive profile and seems to match the ecology that we expect to see. It was a little bit less informative about dive duration, like how long they were diving. It was harder to see the differences between the dive profiles for dive duration. But there was still some trends. Again, the orange dives tended to be a little bit longer than the black dives. These, of course, they matched the centroids for each cluster, matched the dive profiles that we see for other leatherback populations, which is really good. And the ecological relevance, it breaks down to these different groups. So confirming these kind of previous findings that have been put out for this particular population. So I do have some, future work that I could do for applying for future work in the dynamic time warp clustering. There is a predict function that comes with this. The predict function essentially allows you to train your data train your model on one type of, on part of your data, and then predict based on another grouping of data to see what the deviation is. So essentially, I could train my model on one particular year and identify the main centroids for that year. And then I could predict it on another year's data and see how well those dive profiles match. And that would be kind of interesting for this population because they are in the Pacific Ocean, which experiences El Nino. El Nino is a climate phenomenon which actually affects the oceanography in the area quite a bit. So they tend to have different behaviors, and their prey tends to be distributed differently during El Nino years. So that would be kind of interesting to see how the El Nino years differ from the other years. And then, of course, there's a possibility of applying this to other leatherback populations. It'd be really interesting to, potentially also get some data that has actually been ground truthed and see how well that these models predict the number of clusters, for example. I could use the external method for that, of seeing how well they predict. And then the final next step, this is a chapter in my dissertation. So the big next step is actually to integrate these dive profile classifications into a hidden Markov model. And I'm not sure if you guys know what that is, but behavioral models have become quite popular for movement data sets. And hidden Markov models are one of the ways that you can try to determine different movement modes for large data sets like this, for satellite data collections for migratory species. So, the goal with these models a lot of times is to try to bin and classify where these animals are doing foraging behaviors or residential behavior, and where they're doing transiting behavior. But there are, multiple different types of behaviors that you might be able to identify with these types of models. So far, hidden Markov models have been kind of restricted, and they haven't really been implemented in a kind of a large way in RSTAN yet. So I'm really interested in trying to fit these models in RSTAN because RSTAN is very fast. So essentially, I would determine the count of each dive type for each individual per day and try to see if there's differences in distributions for them based on the different states that we'd see. So for example, residential versus transiting. So kind of building on that spatial summary. So with that, I just want to acknowledge my collaborators in this project. Of course, Dr. George Schillinger and published the data. as he is the turtle expert on this. He runs this organization Upwell. So if you guys are interested in sea turtle stuff, especially leatherback research, check out Upwell. And my other collaborator, Mike O'Brien, was super, super helpful in helping me apply our code and expertise. And there are multiple other examples of applying dynamic time warp clustering. So he actually worked on a different type of application of dynamic time warp clustering to another ecological species. This one was for fish. And so, you could check out their paper for another example of this type of clustering technique. So with that, I'll take any questions. I do hope, here's my email. I do hope to publish the code and stuff like that after publication. I think that it could be really helpful for other people that are going through dive time series data or time series data that's kind of similar.
Cool. Great talk. It looks like you have a question already. I was just about to mention the hidden Markov models. I think that's a really good way to go with the behavioral states, especially if you can incorporate covariates like maybe the ocean current, the depth, and the chlorophyll A productivity, so you can kind of get a feel of the energy consumption of the animals as well. That's really, really cool stuff.
Thank you. Yeah, I would like to try to. I'm not sure if I will incorporate the environmental covariates too much for my actual chapter, just because we've already kind of. I don't know, George's paper, and then my advisor, actually, Helen Bailey, has done some work on looking at the environmental covariates for different states. The part that's really been left out for this particular population is the vertical component. No one's really integrated the vertical component into movement models. But yeah, I would, maybe if I have time, I would love to throw the environmental covariates in there.
Do you see the question in the chat box?
Oh, sorry.
What about clustering your profiles to the nearest of those 8 established dive profile types? Would that be fast? Or
classifying to the nearest of the, it's using my data to create the clusters, but all of those dive profiles are, well, most of them are represented already in my data. So I wouldn't want to, I could, maybe using the predict function, what I could do, which might be interesting, is simulate some data. I could potentially simulate, different dive profiles that are known, all 8 of those, and I could use that to predict, using the predict function, predict on my data and see if they match. That might be an interesting idea.
There's another question in the Q&A box, and well, they're saying thank you, and I joined this a bit late. Anyways, how difficult is it to compile satellite data and the right cluster when there is 2 options? Why do you choose 4 and not 6?
So I did go through that in the slides, so it might be helpful to go back through the recording if you want to look at that in detail. But essentially, I expected to see anywhere from 2 to 6 based off of previous published literature. But I used the cluster evaluation indices, which are a little bit subjective, but they're kind of like AIC criterion, if you've ever used that before, to try to determine what the best cluster size would be. And I chose 4. The cluster, the indices essentially told me that that the optimal number would either be 4 or 6. And I went through and evaluated the profiles and the centroids that each one of those gave to me. And 4, I think, makes the most ecological sense, and is probably the best bin of dives for this particular population.
Any other questions? Questions, comments, anything? give them a little bit to think.
Yeah, I was really excited to this was actually quite fun to go through and apply. I think this package is kind of new, so there's definitely a lot of probably different areas you can apply this, and it's kind of fancy because, you know, everyone gets stoked about machine learning, right? And like anytime you talk about any kind of machine learning, so the fact this is going through like in an unsupervised way to cluster things is kind of new and fancy.
Yeah, and the way you explained it is really, really comprehensive too. So I appreciate that. I put the link in the Facebook group, so some people came in halfway through into the actual Zoom thing. So I'll have to post the full video on there later, and everybody can ask you proper questions after they review the whole talk.
Yeah, sounds great. Yeah, anyone, you know, that sees this also, I did put my email up there, so feel free to email me if you have any questions, or if you decide to apply this you know, particular package to your data set and need any help with your code. Yeah, I got help with my code, so I'm happy to pass on that help to someone else.
All right, so I think if everybody is good on that, we can move on to our next speaker. So our next speaker is Julian Perez-Correa, sorry, from, I believe, Universidad Espiritu Santo, Ecuador, right? Are you ready, Julian?
Hey, hello everyone. Yes, I'm ready. Just let me share my screen. Just 1 second, please. Russell, it looks like you have blocked like the option to share my screen.
My bad. I need to make you a co-host. There we go.
Okay, no worry. Hello everyone. Can you confirm that you can see my presentation?
Yep.
Okay, cool. Well, it's a pleasure to be here, and thanks for the invitation to give this talk. I'm gonna have like a presentation about my work in the Indian Ocean and how we find that climate oscillations and invasion of alien species influence the oceanic distribution of seabirds. This work was published last year, like 6 months ago, but however, we still think we can improve some information. So I would really appreciate if you have any observation on this, okay? So this is a work that we did together between the ZESL, Imperial College London, Espiritu Santo University, University of Western Australia, and University of Exeter. And basically, why we think we need to do this work. First of all, seabirds are top predators. That means that they're in the apex of the food web. And also, these species feed over 7% of ocean productivity. This is particularly important because it goes to in a competition with global fisheries. Another important thing is like seabirds has unique traits that makes them evolve to be particularly adapted to live in the ocean. In general, they have salt glands, they have wings with long distance travel, so they have adapted wings, and also they have a modified vision to locate prey in the ocean. And finally, another important thing about seabirds is like they do have important ecological roles. These ecological roles basically function as a link between the sea and land. So by predating fish and other species in the sea, they put together nutrients, they pack together nutrients to the land, and this functions as an enhancer of nutrients in between their colonies, especially. However, despite their importance in the worldwide food web, the problem is that seabirds has a lot of threats in general. So this is a list of some of them. I think those are the most important. So basically, they have a lot of problems: entanglement in fishing gear at sea, Also, there's some overexploitation of seabird prey via fisheries. So as mentioned earlier, the problem is that it goes entirely in competition with fisheries. So when fisheries predate over some resources, seabirds also have a decimation on the prey. Another important problem is climate change. I think like everyone knows about what is climate change and how it may function as a dispersal, as like threats for particular productivity in the sea. Another problem is water pollution, which we all know what problems are with water pollution in general, not only with oil spills, but also with now microplastics that has been like discovered. Another problem is with energy production, and finally with invasive species. And this one, invasive species, I think is one of the problems that has been also well studied in the land, however, not in the ocean. And that's why we need to figure out some more information about this. In general, like seabirds are two main characters about the distribution. They are related to oceanographic conditions and geomorphic characteristics. These two conditions are basically given because they define where productivity and seabird prey are located. And also, because of this, seabirds are good indicators for long-term changes. So, for example, this is a study done by Laura Banocchi in 2014, and particularly in the Indian Ocean, and she found some like relations in between different covariates or environmental variables and different groups. So she found that basically specific seabirds related to a specific energetic relation has different needs on what prey do they need. Yeah. Again, knowing that there are a lot of problems with seabirds, also you need to know that they are really vulnerable to changes and human impacts. And Palexny in 2015 found that approximately 69.7% of all seabird populations, so this is a big number, show a declining trend. And this was found in 2015. And well, we think that this pattern remains the same with more decimation about seabirds population. So another important thing to think about why to study seabirds in general. Okay, we know about seabird problems. However, we need to know also that oceanographic conditions where seabirds are related are also influenced by other mechanisms. These mechanisms can be related to climate oscillations. Actually, in the early talk with Nicole, she talked about the El Niño oscillation, and actually this climatic variation changed the whole food web around the world. However, the problems is not only related with the El Niño oscillation, which it happens in the Pacific Ocean. There are another different oscillation that has been discovered just yet, 20 years ago, which is not that long, and hasn't been studied that much, which is the Indian Ocean dipole, and is measured by the dipole moit index. In general, the mechanisms of this oscillation is like similarly to what happened with the El Niño, when it is warmer in the west, is colder in the east, and vice versa. But also the problems of this fluctuation of temperature may influence in productivity and also in wind and rain. So I don't know if you remember, like last year, 2020, there was some fire in the forest of Australia. Well, this problem actually is related to this Indian Ocean dipole index. And as you can see in the picture, well, it basically fluctuates severely between east and west, sorry. Okay, and another important thing to include in this, this background is like seabirds live in Iceland generally, and islands represent a natural elevators for seabirds because they choose these areas for breed and for rest. So depending on the season, they may use this island for both things. However, as you know that there are a lot of invasive species in some islands that have a predatory effect. One of the main predatory species that are invasive in islands are rats and cats. However, this study will focus particularly on the effect of rats. So these predatory effects makes seabirds vulnerable and reduce the population really, really fast. So they are vulnerable, and this vulnerability of seabirds depend upon species. Some species, especially those ones that nest in burrows, are more vulnerable than other ones that nest in trees, for example. For example, studies done by Graham in the Indian Ocean found that another important thing of seabird colonies is that when there are non-invasive species or small amount of invasive species that predate on seabirds, the areas surrounding these islands are more productive, and actually density of seabirds can go up to 760 times greater than seabirds than islands where seabirds are decimated by rats. So basically, it's important to understand that seabirds population enhance productivity, and when they are impacted by invasive species and by climate oscillations, there may be some effects on these areas. So knowing that the effect of rats and invasive species actually is well understood in land. However, no one has explored area before the effect of rats and climate oscillations, considering the seabirds distribution at sea. And particularly important is to mention that this study was run in the British Indian Ocean territory, because we also want to fill some gaps information of seabird distribution. So what were the objectives of the studies? We have three main objectives. The first one is to model seabird distribution using oceanographic, geomorphologic, and climatic oscillation variables, which is like the broad one. However, we also wanted to model rat invasion sensitivity on seabirds at sea distribution, and finally to estimate potential increase of habitat use after theoretical rat eradication program. So basically what we wanted to do is first of all model seabirds, then model the effect of rats over seabirds, and then try to figure out what would happen if there's a theoretical eradication of these invasive species, and why we choose this area. Well, first of all, because we got data from there, and also it's particularly interesting that the Chagos archipelago in the British Indian Ocean territory is located just in the middle of the Indian Ocean. So basically, we can first of all find out what differences are between the Indian Ocean dipole. Second of all, it is really interesting to know that there is a set of 55 islands where basically half of them are invaded and half of them are non-invaded. So we have a mixture of these effects that can be explored. Another important thing is like as it was a really like broad range experiment, because it has been non-take marine protected area since 2010. So basically there is no other variation of impacts done in the moment. So this leaves us really like open, I would say open, they give an opportunity basically to do some good important things. Also, this area is an important, it has 10 important bird areas, IBAs, that are considered because there is a good population of birds that can be tested. And finally, it has 18 rated seabird species. So all mixture of these characteristics of the area leave us a good like broad range experiment. So the area was sampled between 2012 and 2016. So you can see like how many samples have been done there. So basically, the sampling method was running in a patrol vessel, which every time that there was a transect of 30 minutes running at 12 knots, and all seabirds around a 118 arc were counted and identified, approximately up to 300 meters. And basically each transect has a longitude about 11 kilometers. So another important thing is like the same observer were running all the research between 2012 and 2016. So this reduced particularly the amount of bias provided by having different observations. Unfortunately, I never went there, so the observations were run by Pete Carr, which is another a good researcher that is working in the Indian Ocean. Okay, so we run like a bibliographic research about which variables we think we need to include. And we came with a set of variables that were distance to coast, slope, turtle A concentration, sea surface temperature, sea level anomalies, years, dipole mode index, and oceanic Nino index. Why we think of these variables? Well, because first of all, the first three geomorphic variables, slope, seabed, which is bathymetry, and distance to nearest islands or nearest coast in this case, are particularly important to explain like the geographic distribution of seabirds, considering these physical variables. On the other hand, it is well known that at least sea level anomalies and chlorophyll A concentration are really important to explain distribution, because this one can be used as a secondary secondary result, or yes, secondary result of how seabird prey is located around sea. And we, as you know, seabirds basically needs to seabirds are located where their prey is. And finally, in order to include like or try to test the hypothesis that climatic oscillation variables are also important for seabirds, we include indexes that downloaded from the NOAA, which are the oceanic Nino index as a measure of ENSO oscillation, El Nino Southern oscillation. Then we find the dipole mode index as a index of the dipole, Indian Ocean dipole, and here in order to try to catch up if the variability given of all of these information was like promoted by the year of the sampling, or it was basically a result of some index. So we set out all of these variables, and we use a BRT, a boosted regression tree model, a BRT, which is a particularly interesting model that allow us to have micro regressions and boost them to find which, like the probability of which of the variables are more important. Why we choose this one? First of all, because it is important to know that in general, ecological relationships between dependent and independent variables are usually non-monotonic. That means that sometimes we will have some problems finding like a perfect linear regression, and you know that's why in general there is a lot of methods that have been developed in order to try to find out what would happen with ecological relationships. And I think the BRT models are really important. So we choose this one and use the R package EVM and adapted the code from Elite et al., which she has a really comprehensive like set of coding that help us to understand and to do these models. We run model with all variables, and for all the most abundant species, with those one that has over 100 observations. And then after this first model, we took out those variables that would have over less than 5% contribution in the model, so the ones that are less important, and then we remodel again, and after having the result, we plot it in a map in order to find out how it will be the distribution of the seabirds in this area. After that, so I think the interesting thing of the study, using the distance to coast from of seabirds that were observed is important, yes. However, we think that it wouldn't give us like the particular information that we need to understand about how a bird decides to be like in a space, in particularly only because you know when there is a bird in a particular point, there is a space, a distance in between different areas. So as in Chagos Archipelago, there is a set of islands like reparted in different areas where rat-free are right invaded. Probably the seabird also would be closer because of relation of distance to islands that they prefer. So we include this model, we break down models again. However, we change the distance to coast variable, and we calculate using the nearest distance two new variables: the distance to nearest right invaded island and distance. So these give us an opportunity to do two models for each species. And using the Davis test in the segmented package, we also find a broken stick. We did a broken stick regression and find thresholds of the impact of the distance from the coast, in particular if they are invaded or not. And at the end, we have these two models, a model where we assume that all islands are rat-free, then we have a model where we assume that all islands are rat-invaded, and if we just make a subtraction of raster models, we may find what would happen if, for example, there are theoretical rat extirpation around all this area. And finally, we got the results. Basically, we have 7,008 seabird signings in general, and we have more abundant species. Those ones were red-footed booby, brown nutty, white tern, and wedge-tailed shearwater. So these are the ones that are here in the picture. Really nice birds, actually. And in general, well, as you can see, there were a little bit more observations in 2014, and the most abundant bird that were located was the brown nutty in general. However, we decided to do all models with these birds, and we run the BRT models, and this is the results. In general, most of the birds are related to at least 4 variables. Okay, we can see that seabirds in general respond better to climatological conditions, so that's important to understand. And there is an important distance to coast relation, which was expected because the bibliography says that in general, seabirds and seabirds in general are related where the colonies are located. So we expect to have that information. However, when we go closely, we'll check closely like the differences between the responses of each species. We found that, for example, slope was really important for red-footed booby. However, temperature is important for wedge-tailed shearwater and red-footed booby, and brown nutty was positively correlated with curlbill egg concentration. So this last feature, or these 2 last features, were really important to get the next step. So we included in the model 3 variables, as I mentioned, 3 climatological oscillation variables. There was ONI, like Oceanic Niño Index. Then we have the dipole mode index, and we include the year to catch up the variability. And interestingly, we found that these birds that are more closely related to temperature had no effect of year or ONI, but had an important effect of contribution given by the variable's dipole mode index. And particularly, red-footed booby has a response that where the dipole mode index give a contribution of 6.7%, and wedge-tailed shearwater a response with 23.7% contribution from the dipole mode index. So we found that there is some particular response of the birds that are more related to temperature to this dipole mode index. So we found this was really interesting and fascinating to get, because this gave us then some idea about what would happen with the response of birds with climate change, for example. And we model, we run up the models. As you can see, well, this is a model that is that was run in the context pool of the variables and no longer than the distance to avoid like bias of the predictions. And we find in general that, well, there is a great effect of distance coast, as we found. The IVAs were really enveloped in the models, so were interesting also. The areas that are more marked in general are really close to IVAs, which is also expected because in the field we found like more birds around these areas. Another interesting thing is like shearwater show a scatter distribution, and the red-footed booby, white tern, and brown nutty, and densely aggregated by islands. It's also important to know that in general, wedge-tailed shearwater is more pelagic than the other 3 birds. So basically, again, this is an expected result. However, the next result was quite interesting actually, because as I mentioned earlier, we got these 2 models, the rat-free model and the rat-invaded model, and then we find a BRT to understand what are the differences between them, and we got the Davis test to find out this broken stick regression. So basically, these plots that are here are density are dependent plots of the only distance to coast from the three models that I mentioned. And also we include, just for graphical references, the neutral model, that would be the model without considering these rat-free or rat-invading areas. So we found that, again, red-footed boobies and brown nudie respond better to rat-free islands. Basically, what we found is like red-footed booby and brown nudie are closer to rat-free islands. And the difference is like quite interesting. In general, as you can see in the plots, red-footed boobies are like probably 23 kilometers closer to rat-free islands. On the other hand, brown nudie is like over 10 kilometers closer to rat-free islands. Then, what's fascinating is like white terns respond better to rat-invaded islands. So they are more related to areas where rats are present. And on the other hand, wedge-tailed shearwater has no perceptible response. There is no response particularly given by the wedge-tailed shearwater. And what we do have with things like, because as wedge-tailed shearwater in general is particularly vulnerable because it nests in burrows by rats and other predators, just like the coconut crab, the adaptations, first of all, are more related to avoid predators. So when we did the field survey, probably we didn't catch up basically like really, real observations of wedge-tailed shearwater, because they leave the nest in the morning and they come back in the night. However, it's particularly important to understand that there is an interesting signal about an inverse effect of the invasion of species, where birds that are more vulnerable in land are probably less vulnerable at sea. And this is the final model, where we include the rat-free model, which is in the top of the three species that has a particular effect of rats. Remember that I mentioned that wedge-tailed shearwater was no particularly affected with rats at sea. So we have the rat-free model at the top, then we have the rat-invaded model. Then in the third row, we have the subtraction of two models and find out like where would be a more probability to have increasing of birds. And in the third one, well, it's just the 95% quantile, so the areas that are more interesting. So we found that areas that are rat infested can be potentially recolonized, which is interesting. And I think that one of the most important things, like considering all this area and the differences in between the rat-free and the rat-invaded model, we may have an increase of habitat use between 3% to 14%, considering a theoretical rat eradication. So that means that, again, eradicating invasive species are a really important measure to have to manage, especially protected areas and marine protected areas particularly. Areas with closer seabird population have more probability for a faster restoration. That's another important thing that was published by Boxing et al., but we confirm that particular areas with prey populations are more probable to be restored, because colonies can colonize other areas. Finally, if by increasing or by reducing invasive species, we can increase population, and this increase of population may increase around the water productivity, which that may function as an interesting signal for restoration of coral reef. And also, this enhanced productivity may also influence in fisheries worldwide, because as areas are more productive and are less impacted by invasive species, some effects on some positive effects on fisheries can be found. What are our perspectives for the future? Well, first of all, try to find out some similar patterns worldwide. Actually, we're trying to find out if this effect can be analyzed elsewhere. We also want to measure the effect and influence of subsurface predators. So we are now trying to get some data, not only on seabirds, which we now know what happens, but also how the subsurface predators, so basically tuna, dolphins, and others, are distributed, and how they are related with seabirds. by using, well, we also want to use some seabird tags. Actually, now there are some studies where there are colonies of red-footed boobies that are tagged, and we are now like looking for more information. Also using some bait system for fishes, so with camera traps underwater. We also need to measure nutrients at the same time as assessing birds in order to find out this relationship, not only downloading information from remote sensing, which is what we did, but also measuring nutrients in situ and find if these effects are just a matter of chance, or basically could be also uh, um, get it in nearby continental land. Back home, basically, seabird abundance and distribution at sea in the British Indian Ocean territory is driven by geomorphological and oceanographic conditions. Seabirds are vulnerable to climate change and human pressure, particularly invasive species and other effects. The effects of invasive species extend beyond land. In the spatial distribution of seabirds, the effects of rats varies among species, which is really important. Apparently, seabird species that are more vulnerable on land are less vulnerable at sea. Rat eradication programs enhance habitat suitability and quality up to 14%. The eradication programs should be identify where should be like the best eradication in order to minimize the financial problems and maximize the restoration probabilities. And well, these also give us information about why and how seabirds are distributed now and how they're impacted. But these two important features, which are climate oscillation and invasive species. Well, these are the acknowledgement in general. There's a lot of people that were involved in here, particularly the Basil Petrol in the Indian Ocean Territory, and thanks for the foundation, Batrole Foundation, that gave us some fun for this. And that is, thank you very much. So if there is any question, you can follow me on this social media if you like. So where I include some of my research and some birds pictures if you like. Thank you very much again.
Awesome talk. Really, really good. I
Yes,
never knew that there was
my
this
English
issue with
wasn't that.
invasive rats before. So on one of your slides, you mentioned that white terns are not as impacted as the other three species. Do you have any hypothesis of why that may be?
Honestly, I'm not sure about what did happen. Maybe it's because there's some sort of like differences in competitions on the land on the at sea distribution, because white terns are really small, smaller than the other ones that are the brown noddy and the red-footed booby, and probably they're kind of displaced from some feeding areas, and they need to occupy like areas that are not in competition with greater birds. So that's actually what I think, but we need to do a lot of studies to prove this.
Cool, cool. All right, so questions. Do we have anybody? Please write them either in the chat box or the question answer dialog box. We'll give everyone some time to think and process all of this information. Questions, anybody from the 74 attendees? Okay, we have one from Valentina, who says, in Ecuador, there are good places to do that investigation. Or are there good places to do that investigation and compare it to another place also here in Ecuador?
Okay, thanks for the question, Valentina. Actually, yes, we're thinking about using same approach around Galapagos Islands, where basically has similar conditions in terms of invasions, where some islands are invaded and others are not, and also there's some seabird colonies around. However, the problem would be like to catch up the information as we need it. So we are like talking with Darwin Foundation and see, we're in talk actually, and see if there's possibilities to get this.
I had another question too. In the beginning, in your methods, you mentioned that you do transect surveys around these islands. Yeah, and I think you said that you do 300-meter distance and you count the birds and the species. How do you do that? Just by single observer, like slowly going by and counting every single bird that they see?
Well, yes, actually, what the observer did was like going in the vessel and patrol like um, at 12 knots. So this give basically a transit of 30 minutes and approximately 11 kilometers. So basically, there were two observers, the main one and well, the assistant, and both of them were like looking all the birds in this arc of 180 degrees, and basically count all of them. So another important feature that we didn't include in the analysis, but it's also interesting to get, is like there were also some seabird segregation that were found in these areas. However, these seabird segregations are basically given by some feeding areas, and as they're dense in this area, may give us some bias about their distribution. However, we're thinking that's why we think that we also need to include this, but with a more standardized methods, because most of these aggregations were get from by chance and not in particularly included in the transects.
Cool. Sounds like a lot of work.
Indeed, we do have.
Let's take at least one or two more questions. I'm sure everybody has something in their mind. If you came in late, feel free to ask him some questions to extrapolate on what he was talking about.
Well, it's fine not to having question. Maybe no one understood me. Maybe I was well explained.
Oh, I think it was pretty clear. We have one in the question and answer dialog box. Don't you think that it needs long-term monitoring to link invasion with climate change?
Yes, we do think we need some a lot of time in this, because this was only a 5-year survey. However, this gives us a signal. We found something, and we need to keep like doing some research on this in order to find if these trends are particularly true or just was a matter of how like birds were distributed in the area. So I really I agree with your observation actually. But well, this is what we have now, and at least it was what the model gave us. I can hear you, Russell.
Sorry, can you hear me now?
Yeah.
We have another question from Melanie, who asks, are there other alien species besides rats that have influence on the seabird distribution?
Well, in this particular study, no. Okay. However, it is known that in general, other visited species such as pigs, cats, dogs may influence and on the density of seabirds population especially in the breeding areas. And it is known that these invasive species decimate and reduces like also the probability of, of, of growing the next generation. So basically they are a lot of these invasive species, and we need to do some studies more on how they're impacted. For example, I know in general in Guadalupe Island in Mexico, they do have a lot of problems with cats.
We have another couple questions. So from Jason, for the baseline surveys, how was citability differences of different species accounted for?
Okay, well, the observer actually has a lot of experience. Actually, has been monitoring the Chagos archipelago like for over 15 years, and he is really familiarized with like seabird patterns and seabird shapes, colors, and how they move around. So we tried to, or he tried to get all the birds that were counted, and actually some of the birds that were not identified, which are, well, there was just maybe 10 or 20 birds that were unidentified, were taken out from the model.
We have a question from Laura. How do you think climate change will further impact the species distribution?
Oh, because as the climate varies, also this influence on birds prey and on actually ocean productivity. So as the ocean productivity is impacted, the prey of the birds are also impacted by it, moving and changing the pattern of the distribution. So therefore, places that are now currently known to be of good production for like for fishes and shrimps and facets and other like prey, seabirds prey, can be moved. So probably seabirds may need to use more energy, or may need to find some other areas to feed on, and this also can impact in how like prey is getting from. So that's actually one of the main problems of seabirds, and also this problem is related again with the possible decimation of fisheries, because as you know, because as temperature of the sea rises, there are a lot of like changes in the patterns of distribution of the productivity.
We have another one from Astrid. As many species move with increasing global warming, are there any other marine, mammal, or fish invasive species that may be competitors over food sources as the impact of climate change increases? So I guess as climate change impacts start taking place, will there be more invasive species competing in those fisheries or even on the land in the islands?
Okay, that's a very good question. If we consider ourselves an invasive species, well, I can say that we are the best competitors with seabirds, and especially with the first their food source. And however, as like climatic variability increases, also inland, it will be more difficult to get some food in some areas, in particular in islands where are really close. So I think the pressure of seabirds in these areas were increased because there would be more needs of invasive species inland. And I mean, at sea distribution, there are another like invasive species that should be considered. For example, now I'm studying the distribution of the snowflake coral, which is Caricoa rizae, and this particularly like invasive coral is like decimating population of other corals and other macroinvertebrates in coral reefs. And also as this invasive species is increasing population with some like temperature increment, is spreading in some areas, and the spreading of this invasive coral reduces population of the native corals. And this also would mean that areas of feeding areas of birds could be decimated. But I mean, these studies are completely different, and I cannot get like relate them because the snowflake coral research is here in Ecuador, and the other one was in the Indian Ocean. So they're really far away. However, I mean, we can match both knowledge and see that probably not uh, the impact of climate change can gradually also imply some invasions of other species that were not related in the study.
Okay, so I think if that's it, we'll end the talks here. And does anybody have any other questions? Anybody? No. Okay. Well, I thank everybody for coming and watching the talks. Sorry that for those of you who just joined us halfway through the first talk, when I posted the link on Facebook, we had some technical difficulties that hopefully I can get sorted out before the next ones. We will be having our next talk, I think, let me check the schedule, at 6. That is on a different time scale. Let's see. So day 3 will be at 6 p.m. Eastern Standard Time, and that will be on March 4th. Okay, and thank you again, everyone, for coming out. Thank you, Julian, for talking. Your joining us was a lifesaver for me, for sure. I would think our other speaker, but I think she left already. She probably has a pretty busy schedule too. All right, and I will see all you guys later. Bye.
Bye. See ya. Thank you for the invitation, Rizzo. Thank